Welcome back to Acosta's Anatomy. In this lecture, I'm going to discuss hemostasis. So what is hemostasis? Hemostasis, generally speaking, is the prevention of blood loss. So in the event where a blood vessel is cut, an artery is cut, due to whatever type of trauma to the finger, just as an example, red blood cells are traveling through those arteries. So we want to prevent those red blood cells from leaving. And so what I'm going to discuss is the mechanism in which how we prevent these red blood cells from leaving. But the first thing I want to point out here is this model. So if you look at this model, this is what represents an artery. And so a little bit about the anatomy of the artery, this is what's known as the endothelium. So this endothelium is composed of simple squamous cells. So these simple squamous cells here provide a slick layer. So whenever this blood is flowing, it can flow at an efficient rate. And so this endothelium, the next layer proximal to it is this, which is the connective tissue. So you have the connective tissue, which has a lot of the collagen fibers. And then this layer here, this is a smooth muscle layer. So this smooth muscle layer is what can control the diameter of the artery. So whenever this muscle contracts, it results in what's known as vasoconstriction. If this muscle is relaxed, what's known as vasodilation happens. And so with vasoconstriction, whenever it tightens, it reduces the blood flow. Okay, so that's it for this model. These models over here, so this is what's representing a red blood cell. Remember, once again, this is what's traveling within the artery, and it carries oxygen using what's known as hemoglobin. This model is what represents a platelet. And so we're going to discuss the mechanism as far as how these platelets form. But what I want to do is give you a general overview here of the different phases. So there are three main phases for hemostasis. First is vasospasm, followed by the formation of our platelet plug, and then the last is coagulation or blood clotting. There are a couple additional steps. So after we form a blood clot, we don't want that blood clot to stay in there for a really long time. And so, whenever this tissue is damaged, the cells are going to regenerate. So for instance, these endothelial cells, these simple squamous, they'll start to regenerate, so will the smooth muscle. And then the clot is then, it's then broken apart, it dissolves. Okay, but we're not going to get into the details of that aspect, but we are going to go over these three different phases. Okay, so generally speaking, vasospasm, that means that the blood vessel is going to constrict because we're trying to reduce blood flow to prevent the blood from leaving. Second is the platelet plug formation. So all of these platelets, they will, wherever there is a breakage, they will start to accumulate here, right? Because we're preventing the blood from leaving. So they accumulate here, and then the last stage is coagulation or blood clotting. And so the clot that's formed is due to this fibrin mesh, which will anchor this blood clot here and hold it in place while the tissue can regenerate itself. Okay, so that's generally speaking here. So now let's start to get into the details of how all these different things happen. So the first thing I'm going to talk about is vasospasm. So what is the mechanism of that? Well, there are a couple of different players there so that allow this to happen. So one is that we secrete a molecule which is known as endothelin. So endothelin. So endothelin is secreted by these endothelial cells. So it'll get secreted from here. And what it will do is it'll bind to the smooth muscle that's here. So when it binds to the smooth muscle, that is what allows contraction, so vasoconstriction. So let me point out here for vasospasm, what we're trying to do is to reduce the blood flow. Okay, so endothelin is one. Another thing that occurs is the activation of nociceptors. So nociceptors, that means pain. These are pain receptors that are found here within this particular layer. And so in order to activate these pain receptors that are found within this layer, we need what's known as inflammatory mediators. So for instance, histamine is an inflammatory cytokine. Another aspect of vasospasm, it's called a myogenic response. So myogenic response. So this is just due to direct contact or stimulation of this smooth muscle. Just because, remember, once again, blood is flowing out this way, so it's going to activate this smooth muscle. Okay, so that's vasospasm, how the muscle, how the artery is going to constrict. So the next thing is the platelet plug formation. So one thing you should be thinking about, I've already mentioned that there are platelets that are circulating here within this lumen. Okay, so here is a platelet here. But one thing you should be thinking about is, how is this platelet, how does it know where to go? How does it know that there's an injury occurred at this particular spot? And so once it starts to contact this purple structure here, this is what's known as the exposed collagen. So I'm going to write that here in blue. So exposed collagen. So contact with collagen. The platelet. So in order for this platelet to actually bind here through a process which is known as adhesion, it comes into contact with this collagen, it activates it, but it needs a particular type of protein known as the von Willebrand factor. So the von Willebrand factor, this is secreted first off by platelets. So once they get activated, the platelets, they'll secrete the von Willebrand factor, and then they'll also von Willebrand factor was also secreted by the endothelial cells. And so as the von Willebrand factor, as it comes over here, so I'm going to draw it as a triangle here. So as it's secreted and it binds here to this collagen, this is what enables the receptor that's found on the platelet. That's what enables it to bind there. So the glycoprotein, so I'm going to do the glycoprotein in red. So there is a glycoprotein. We're not going to get into like the specific name of it, but we need to understand this concept. So I'm just going to write glycoprotein, and then GP is for short. So the glycoprotein, this is what's found on the platelet. So this is what's found on the platelet. So this glycoprotein that's here, it'll bind to the von Willebrand factor. So once it binds here, adhesion is complete. So after this process is complete, we move on to the next stage. So the platelets, they have contents that are found on the inside, and we call them granules. And so a couple processes need to occur here for the release of these granules. So after adhesion, the next process is recruitment and aggregation. Because we can't just have one single platelet that's here to prevent blood from leaving. We need a bunch of them. So in order for the recruitment of other platelets to get to this particular site, there are chemical messengers that are sent, that are released by this platelet. And so the name of these molecules, so one of them is thromboxane A2, as well as ADP. So once they released, so I'm going to write, so this will be A, and then this will be B. So once it binds and then it releases A and B, so thromboxane and ADP, it'll get to A and B will then bind here at this particular platelet. So once it binds to this platelet, it gets recruited, and then after that, in order for both of these, so let me draw this. So there's one platelet, there's another platelet here. So in order for these platelets, they expressed another, after the binding of ADP, they express another glycoprotein. So glycoprotein. So once again, I'm not going to get into the specific names, but we need to understand this concept. So glycoprotein. Once again, it's a GP. So upon binding of ADP and thromboxane, it gets recruited, it expresses this particular receptor here, this glycoprotein. And then what's also another factor that's secreted by these platelets for aggregation is what's known as fibrinogen. So fibrinogen, that's this protein here, that it's what links these two platelets together. So this process will continue to happen. We're recruiting more and more platelets, and then they start to aggregate, and then they'll kind of form this chain and they'll link together over here. Okay, so these are just three of the contents that are secreted by the platelets. There are other contents that are secreted by these platelets. So an easy way to remember this is cash. So they release calcium, they release ADP, they release serotonin, as well as histamine. So they're going to release calcium because calcium is important once we get to coagulation. So calcium, I'm going to write coagulation. And then ADP, we've already discussed this. So ADP is important for recruitment and aggregation. And the bottom two, serotonin and histamine, both of these help to increase vasoconstriction. Increase vasoconstriction or vasospasm. Okay, so that's for the contents that are released. That helps for recruitment and aggregation. Another factor to consider here, so as a result, when all of these platelets start to accumulate here, they start to have this negative charge. They'll have this negative charge that's here. And so this negative charge it's on the phospholipid bilayer here of these platelets that has this negative charge. And it's due to what's known as phosphatidylserine. So it gives it its negative charge. So this is important because this is necessary for some of the particular steps for blood coagulation. Okay, so that's going to do it for platelet plug formation. The last step here that we're going to go over is the process of coagulation. Okay, so the end result for coagulation, what we get is the formation of an insoluble fibrin mesh. So this insoluble fibrin mesh, that's what's going to be found over here. So it's these proteins here that are going to reinforce our clot, or the blood clot. So the blood clot, there are a couple of different components here for this clot. There's going to be a bunch of red blood cells. There's going to be platelets, and then as well as the fibrin protein for this blood clot. Okay, so we have an insoluble fibrin mesh here that helps to reinforce the clot. So then another result of the coagulation for blood clotting. So after the insoluble fibrin mesh, we're gonna come back to this, but there are a couple of the requirements for blood clotting or for coagulation. So what's required here are what's known as these clotting factors. So these clotting factors, they are synthesized. They're synthesized by the liver. And these clotting factors, they are in their inactive form. Let's see, I'm going to write. They're in their inactive form. Because we don't want clotting to happen in a normal blood vessel. We only want the blood clot to form whenever there is a breakage. So these clotting factors, they are inactive. So then that's what leads me to the result. So the result of this is a cascade. So it's a cascade in which enzymes become activated. So these clotting factors, they are indicated by Roman numerals. And so the naming of this, the Roman numerals, it's based off of discovery. So for instance, factor 1, so factor 1, this was the first clotting factor that was discovered. And then it goes all the way until factor 13. So this was the last one that they discovered. Okay, so another one of the requirements, so one of them is what's known as vitamin K. We also have discussed calcium, right? Calcium is important for the coagulation, and I'm going to explain that a little bit more over here. But so you have vitamin K, the calcium, and then also phosphatidylserine. Okay, so this is generally speaking the result, the requirements. And the next part to discuss is the phases. So there are 3 different phases for blood clotting. So once again, I've already told you that the end result is the production of this fibrin mesh. So we get this fibrin mesh over here. But how do we get to this point where we have this? I'm just going to leave that here. How do we get to this point? Well, so you go through what's known as the extrinsic and the intrinsic pathways, but what they'll lead to, in order to go through to the next phase, to phase 2, you need what's known as factor 10. So factor 10 in its active form requires a cofactor known as cofactor 5. So you need 10, cofactor 5, and then you also need calcium. So this is a specific example. And what will happen, we get the formation of an enzyme which is known as prothrombinase. So prothrombinase is a complex that contains active factor 10A, factor 5A, as well as calcium. And once we have prothrombinase, it's also known as prothrombin activator. The reason they call it that is because it converts prothrombin into thrombin. So what I'm showing here, that prothrombinase, that's the enzyme that's converting prothrombin into thrombin. So this is just the name for it, but the Roman numeral for it is number 2. So when was this discovered? This was the second clotting factor that they discovered. Okay, so after prothrombinase converts prothrombin into thrombin, thrombin is what's going to be used over here. It's going to come over here, and it's what's going to convert fibrinogen into fibrin. So fibrinogen, that's the first clotting factor. So once we convert fibrinogen into our fibrin mesh, we now have completed the coagulation process. We have this full blood clot that is formed here. Okay. All right, so that's going to do it for this lecture.